share mode. And, okay. Okay, just to confirm very quickly, this is the top part of the slide, the
Yes, sir.
pointer. Bottom, can you see?
Yes, sir.
Pointer here.
Yes,
Okay.
sir.
Left
Yes, sir.
and right.
Yes, sir.
Yes.
So good morning, everyone. Saturday morning. I think last time, as we had started talking about the transcription process, uh, we had initially talked about what is the central dogma. Then we talked about the difference between DNA and RNA in terms of what bases are present and the difference in the sugar portion and how that led to, in a sense, instability of RNA. We also talked about the different types of RNAs which are present typically in a cell. The main one being tRNA, mRNA, and rRNA. And then we started looking at these enzymes which are called as polymerases. Okay. More specifically, these polymerases are DNA-dependent RNA polymerases, which means they will read DNA as a template. The template is actually read in the 3' to 5' direction, and the RNA is actually made in the 5' to 3' direction. Okay, and because RNA contains adenine, guanine, cytosine, and uracil, the nucleotides which are required for polymerase to work are ATP, GTP, CTP, and UTP. Okay, the main thing is they do not actually need a primer and they can start the synthesis on their own. Okay. The polymerase itself, as we talked about last time, is multimeric and contains 6 subunits, out of which 2 subunits are identical, which are called as the alpha subunits. So that's why they show it here as 2 of this alpha. Then there is a beta subunit, which may or may not be identical. Usually it is not identical, so shown as B and B prime. And then there is a omega subunit and a sigma factor. Okay. Now these 5 are always there as part of the RNA polymerase, so that they are called as the core units, and they form the core enzyme. While this sigma factor is not always associated, but it is needed for the polymerase to begin its work. That's why when it is there, the enzyme is fully functional and it's called as a holo enzyme. Okay. Uh, can you hold for a second? I'll just be back. Just.
Yes, sir.
Sorry for that. There was a background noise. I try to take care of it. Okay. Everyone okay? Everyone can hear me correctly? Hello?
Yes, sir.
Okay. So as I was saying, all 6 subunits are needed for the enzyme to function completely as what is called as a holo enzyme. Okay. Now more specifically, the functions of the individual subunits are listed here. Alpha and alpha subunits, they are the ones that detect the DNA which has a coding region. Okay, so they locate the DNA which needs to be transcribed. Okay, B prime binds and opens the DNA template, causes unwinding, and B actually does the polymerization. Okay. Now the main role of the sigma is to recognize what are called as the initiation sites, or what are called as classically speaking promoter sites, based on the presence of these specific sequences. Then actually the transcription will begin. Okay. This subunit, which is the omega subunit, is always found associated, but the specific function is not yet been elucidated, but it is always there. Okay. Now, what are the roles of all of these subunits when they are present together? Okay, which means what is the function of the actual RNA polymerase complex, as it is called. Okay. Now here are the 4 or 5 things it does. Okay. So the first thing it does is it searches the DNA and finds the promoter site and binds to it. Okay. So one of the functions of this complex, or what is called as the RNA polymerase complex, is to find where the promoters are, detect it, and then bind to it. The second thing it does is, after binding to this region, it unwinds a short stretch of double helical DNA. Okay, because DNA is normally present as a double helix. And then at that portion where the DNA has been separated, it reads one of the strands as a template and starts making the complementary what is called as a polymer. But this time the complementary polymer uses what are called as ribonucleotides. Okay, so it starts forming what is called as RNA, and it takes one nucleotide at a ribonucleotide at a time and catalyzes the formation of the phosphodiester bond. The general reaction is shown here. So if I have a piece of RNA already there, and a new RNA nucleotide triphosphate comes in, complementary to the base which is present in the DNA template, then that will be added on to the RNA. I'll get a longer piece of RNA, and this pyrophosphate which is released will further ionize to 2 inorganic phosphates, and that drives the reaction in terms of the thermodynamics. Okay. Now, as it starts reading the template and making the polymer, at some point it will reach a point where it will detect what are called as the termination signals on the DNA. As soon as it detects that, it actually stops the production of RNA, and the RNA simply leaves the complex. Okay. In addition to this, okay, it also will interact with a lot of other sequences and proteins which also play a role in the regulation. For example, what are called as activator proteins and repressor proteins. Activator proteins are the ones which interact with RNA polymerase in a fashion where it makes polymerization even more efficient than normal. And then repressor is where, when it is present, the RNA polymerase is unable to do its function of synthesis of RNA, and therefore it inhibits the production of RNA. Okay, so these are all the functions of the RNA polymerase. As I said, it actually works very similar to DNA polymerase. Okay, so if this is a DNA template and there is a specific base present, then what it does is it picks a complementary base which is actually present as a triphosphate, and then which is present as a triphosphate, and then joins them together. Okay, so for example, let us say this is the RNA which is already there on the template. This is the new base that has to be added. This base is red. Let us say it is A, then here it will be UTP. If it is G, then it will be CTP. Okay, and once this sits in place, then the 3' OH attacks this phosphorus, releases a pyrophosphate, and this one becomes joined to this polymer. Okay, so even RNA has polarity just like DNA. It has got a 5' end and a 3' end. Okay. Now, as I said last time, and when we discussed, this RNA polymerase has got no pre-feeding capacity. So if it does make a mistake, it stays as is. But it has got no grave consequences, because this RNA, after being translated to proteins for a certain time, will be degraded, and this mistake of the RNA will be reflected in a few protein molecules which are made incorrectly, but these are not heritable traits. Okay, so And remember, when transcription is occurring, not only one strand of RNA is made, but multiple strands of RNA are made, and most of them are made with fidelity. Okay, so if there is one error here or there, it will be a individual RNA strand which has got an error, but not all the RNA molecules. Okay. Now, the big difference is that when we look at prokaryotes or bacteria, which are very simple organisms without a nucleus, vis-a-vis eukaryotes, which are very complex. They have cell organelles, nucleus, etc. What we find is that in bacterial systems, all the three classes of RNA, that is rRNA, or what you call as ribosomal RNA, messenger RNA, and tRNA, are all made by a single RNA polymerase. Okay, and this is called as RNAP. Okay, RNA polymerase. In eukaryotes, what is observed is that each class of RNA is actually very specifically made by a different RNA polymerase. Okay, so there are at least 3 RNA polymerase. One RNA polymerase 1, which is involved in making what are called as ribosomal RNAs. RNA polymerase 2, which is involved in the synthesis of what is called as messenger RNA, which eventually will be translated to give you proteins. And RNA polymerase 3 is the one which is involved in transcribing DNA to give transfer RNAs and small ribosomal RNA. Okay. Now, so the big difference is in eukaryotes, we have specific polymerases to do specific synthesis of RNA, while in prokaryotes, all the RNAs are made by the same single type of RNA polymerase. Okay. Now, the process of transcription itself, now that we have seen what is RNA, what is DNA, what are the different types of RNAs, what are some of the properties of RNA polymerase, the actual transcription process and we start looking at it by in prokaryotes, has 3 stages. Okay, and these 3 stages are also present in eukaryotes. Okay, what is called as initiation phase, elongation phase, and termination phase. Okay, this is very similar to what we saw when we looked at DNA replication, where we talked about initiation occurring at the origin of replication, then elongation occurring, and then termination occurring when the replication forks meet or when you reach the end of the DNA. Okay. So, in transcription though, the initiation phase is a phase where RNA recognizes the promoter, opens up the double-stranded DNA, and installs the first base of the RNA. Okay, so what is called as the start of transcription. Once this is done, then all that happens is as the RNA polymerase moves further, on the DNA template, the DNA ahead of it is continuously unwound to give single-stranded DNA, and the RNA polymerase can then nicely read the template and incorporate the corresponding RNA nucleotide, and the RNA strand will be made in the 5' to 3' direction. Okay. As the RNA polymerase goes along the DNA template, at some point it will get to what are called as signals, which indicate to the RNA polymerase that it is time to terminate the synthesis of RNA, because you have reached what are called as termination sequences, and at this point the RNA polymerase will stop the synthesis, and the RNA which is produced is separated from both the RNA polymerase as well as the DNA template and released, okay, for whatever processing that has to occur or whatever the fate is after that. Okay, so let us begin our discussion with the initiation phase, where we talk a little bit about promoters and how they help in the start of the transcription process. Okay, so in prokaryotes, as I say, the promoters are actually sequences which are about 40 base pair long, and they are on the 5' side of the transcription start site. Okay, so So if I were to look at double-stranded DNA and I write the DNA, and when I write the DNA, I'll always be writing one end as 5 prime and the other end as 3 prime, and the opposite side will be exactly opposite. Where there is a 5 prime end on the first strand, there will be 3 prime end on the other strand, and where there is 3 prime end on the first strand, the corresponding double-stranded DNA will have the 5 prime end, because DNA is anti-parallel. So promoters are sequences which are about 40 base pair in terms of the length, and they are on the 5 prime side. Okay. Now I'll be more specific what do you mean by the 5 prime side after a couple of slides where I actually show you how these promoters are set up. Okay, so for now just remember they are going to be on the 5 prime side of where we are going to start making the RNA from, and they are about 40 base pairs long. Okay. Now what you find in this 40 base pair region, you find what is called as two consensus sequences. Consensus sequences are sequences which are seen conserved, okay, across many bacteria. Okay, and if you look at several bacteria and their promoters, you'll find a sort of identity with respect to this sequence across the several genus and species. Okay. And in this case of prokaryotes, what you find is that in the -35 region, as it is called, there is a sequence which reads TTGACA. Okay, and I'm talking about the sequence as it is present on the DNA. Okay, that's why it is T. Otherwise, it would have been U, right? So this is a sequence which I'm talking about on the DNA, which is TT-GACA sequence. Okay, and in addition to this, there is also a sequence which is referred to as a -10 region, and which is called as a Pribnow box. Okay, Pribnow box because of the person who discovered this called Pribnow. And here you will find a sequences which goes somewhat like this: TATA80. Sometimes this is also called as Tata box. Okay, Tata box or Pribnow box. Okay, and this is present in the region which is called as -10. Okay, and as it says, this region is ideal for unwinding. And the reason is ideal for unwinding. Any idea why it is ideal for unwinding?
So in that DNA replication, we have seen the 13 base pair sequence. So that we discussed there ki if there is more number of A and T, it is easy to unwind as compared to more number of GC base pairs.
Exactly, exactly. So see, if you look here,
Because
the
there are
entire
only
thing is
2
80.
bonds.
Yeah. Only 2 hydrogen bonds. Okay, so this region is ideally set up for DNA to be separated with more ease than if it were all GC. Okay. Now, as we look at this promoter, here are some properties of promoters which are important to keep in mind. Okay. Uh, the closer the match, okay, to the consensus, the stronger is the promoter. So what it means is, uh, if I compare the sequence which is present in a given bacterium, okay, to the sequence which is considered to be the wild type sequence or the most commonly occurring sequence, the more closer a bacterial promoter is to this what is called as a comparator sequence or the target sequence or reference sequence, the stronger will be the promoter, which means transcription will occur more easily and at a faster rate and at a faster pace. Okay, uh, and the more the deviation from the consensus sequence or what is called the base sequence, the slower will be the rate of transcription or more sluggish will be the transcription. So the amount of RNA you will generate will be less in terms of number because you're not going to read it as often or as quickly. Okay. The absolute sequence in the spacer region. And what do you mean the spacer region is? We have talked about the -35 region and the -10 region. In between that, let us say the -20, -25, there whatever is the sequence is of not that much of a consequence. What is important is what is the sequence at that point, which is about -35 and -10. Those sequences are important. Intermediate sequences are not that important. Okay. Now the length of the spacer is important. Okay. So for example, if I have a longer spacer such that the gap between these two is no longer 25. Okay, for example, one is -10, and because there is a big gap, this ends up to be -40, which means I put 5 more bases. That can have an impact. Okay, so the length of the spacer is important. Okay, so see this, I have TTGACA, which is, if you remember, the -35 region, and then in between it should be 16 to 19, and then I should see the Tata box. Okay, so if this 16 to 19 is violated, so say it is 14 or it is 21 or it is 13, 22, then I'll find that the way the transcription begins is affected and the transcription is slower. Okay, and it can be as strict as even 15 to 20 could be bad. Okay, so spacers that are longer or shorter than the consents make for what are called as weak promoters. Okay, so here is what I meant to say. Okay, so here we have, if you look at this Tata TACTGT. Okay, what is called as a Pribnow box, or what you call as a Tata box, and here is that -35 region, what is called as a TT GACA region. Okay, TT GAC. Okay, so what is important is in this region, the closer you are to TTGACA and the closer you are to TATAAT, the better you are. Okay, and again here see this gap, they put 11 to 15, actually it should be anywhere from 16 to 19. Okay, now look at this gap here, there's another spacer here, and then based on this gap here, this is what you call as a transcription start site. Okay, usually the initial site as it is called. Okay, now so here is shown this sequence of what is called as -35 and the Pribnow box with respect to several different what is called as genes. Okay, so what is important is genes whose sequences are closer to the Tata GACA box will be transcribed with more efficiency than any other sequence. Okay, so look at, let us say TATAAT. See TATAAT. See this? This is almost identical to this sequence here. TATAAT. All these others are off by 1 or 2. Okay. The more off you are, the slower you are. And here I want what is called as TT GACA. Okay. See this? I see TTGACA. I see again here no TTGACA because it says a TTACAG. Okay, so TT again GACT. Okay, so TT. So I have to keep looking at this region and see how close I am to this Tata GACA sequence. The more closer I am, the better I am. But in general, you'll find the sequence here, the sequence here, I have a lot of match, and here invariably it is rich in TA and T. Okay, so what is important to note is that the closer you are to the sequence, which is Tata, and the closer you are to the sequence TT GACA, the better you are, and this spacer here should be around 16 to 19. Okay, and based on this sequence, the RNA polymerase which comes in will read, okay, oh, this is a -35 region. Then it looks at this Tata GACA region. It starts separating the. It will actually start the synthesis of RNA at what is called the +1 region. Okay, so this numbering is very interesting. So this is called -10 and -35 because actually transcription begins here. Okay, so if this is +1, this T here will be called as -1, A will be -2, C will be -3, the next C will be -4, then -5, -6, -7, -7, -8, -9, -10. Okay, there is no 0. Okay, so it is not +1 then 0 then -1. So +1 is from where transcription is going to begin, and then to the left of that is all minus. Okay, so that's why these are called as -10 region, and this is called as a -35 region. This is all relative to where transcription is going to begin, which is called as the +1 position. Okay, everyone clear here? Everyone clear?
Yes, sir.
Yes, sir.
Okay, So now, as we go further, I'm going to tell you some terminologies which are used when people talk about RNA synthesis, and sometimes it can be a little confusing, but you need to keep that kind of state in your mind. Okay. Now remember one thing, and this is where things get confusing. This sequences which we talk about, TT, GACA, and TA, 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 are the sequences which are on the 5' side of this initiation start site. Okay. So, which means that the orientation of this strand here is actually 5' and 3'. Correct? That's why I'm saying that this is on the 5' side. A couple of slides ago, I told you that RNA polymerase, okay, always synthesizes in the 5' to 3' direction, but more importantly, it actually reads the template strand in the opposite direction. Correct? See, the DNA template is read in the 3' to 5' direction, and I am going to make my RNA in the 5' to 3' direction. Okay. So if you were to go back to this, you will realize now that even though I call this as a transcription start site, when the polymerase actually starts making the RNA, it is not going to read this DNA. It is going to read the DNA which is actually complementary to this. So the DNA complementary to this would be, for example, containing T, GG, GC, AAAAA. It's going to read that and make the RNA, and it's going to make the RNA in this direction. Okay. So it's very interesting. The RNA polymerase comes from here, it reads this sequence, then it reads this sequence, then it starts separating the DNA, it goes to this point, but it doesn't read this as a template. It reads the other strand as a template and continues making the RNA in this direction. Okay. So actually I can even tell you what RNA it will be making. The RNA the polymerase will make will have the sequence A, C, 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 G, U, 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 U. Why? Why did I say that?
So because the other strand is complementary and mRNA will be
Exactly.
produced complementary to that.
Exactly. So if I'm reading the other strand, I'm going to make the RNA which is complementary to that, which is exactly this sequence. Okay. So are you seeing that? So the RNA I'm going to make will be exactly containing this same sequence, except where T is there, there will be U. And the reason for that is because I'm reading the other strand as a template in the opposite direction. And the other strand ka orientation, if you remember, is 3' to 5' in this direction. Okay. So I'm actually reading the strand, making it in the 5' to 3', but reading the strand in the opposite orientation. Okay.
So it's
Is that
like
clear?
it is adding the nucleotides in 5' to 3', but it is reading in 3' to 5'.
Correct, correct. Because the template is always complementary, no? So if you look at this, see, the template is in this orientation, correct? So if I'm coming here and I'm starting to add, and I'm adding say U, and then I'm adding U, here it should be T and T, but in the direction which is 3' to 5'. Okay? So that complementarity as well as anti-parallel nature is maintained during the polymerization, and this is the same as what happens in DNA. If you remember, in DNA also exactly the same. The strand which is read and the strand which is made are always in the opposite orientation. Okay, is that clear, everyone?
Yes, yes, sir.
Does that make sense? Okay. So with that,
Yes,
now
sir.
let us look at this slide. Okay. So this is what I was talking about. This is the double-stranded DNA. Okay. This is the double-stranded DNA, and let us for a moment presume that your Tata box is somewhere here. Okay. So when a Tata box is somewhere here, the RNA polymerase comes in, and then it starts to unwind the DNA, and it's unwound the DNA and has reached this point. And now see what is happening, it is reading this strand. And what is the orientation of this strand? See this 3' to 5'. It's looking at this strand and is making the RNA and the RNA is made in the 5' to 3'. So if you look at the RNA now and look at the sequence here, see it will be complementary. Okay, see this? Here is A, here is A. Here instead of T, here there is a U. There is a G, here is also a G. There is a G, here is a G. So this sequence here is actually identical to this sequence except wherever there is a T, there is a U. And the reason for that is it is reading this strand as a template and simply making the strand complementary to this, which has got the same sequence as the other strand of DNA, okay, which got separated. Okay. Now, because of this unique feature here, that this RNA which is made has got actually the same sequence as the DNA strand which is not being read as a template, this strand here has got interesting nomenclature. The first thing you call it is it's a non-template strand, okay, which is very easy to understand because I'm not going to use that as a template. In the same way, it's very easy to understand that this is a template strand, okay, because I'm reading that as a template. But these other two terminologies are sometimes confusing. The non-template strand is also called the coding or the sense strand, okay. And the reason for that is that its sequence is identical to the sequence of RNA, which is actually going to make sense to the ribosomes, who are going to read this and make the protein. Okay, so this sequence is called the sense sequence because it is making sense to the ribosomes and is translatable. And since this DNA has the same sequence as this RNA, this DNA is also called as the sense strand or the coding strand. And in that regard now, the template strand would be called antisense strand or the non-coding strand because it actually doesn't make sense to the RNA, what do you call it, the ribosome machinery, which is going to make protein. Okay. Now, please remember, this is very important to keep straight, okay, because here and there people will use this terminology of coding strand, antisense strand, etc., etc. So, you have to remember, whenever we say sense strand or coding strand, it means it has got a sequence which matches the sequence of the RNA, okay, which is translatable. And whenever we use the word antisense or non-coding, it means it has got a sequence which is actually complementary to what makes sense, which means it is not going to make sense, and that's why it is called as antisense. Okay. Now, as this RNA comes out, because it is going to make sense, then this is the one which is going to be read as what are called as those codons. If you remember, AUG is start codon, then GCA is the second amino acid, UGC will be the third amino acid, and so on and so forth. And this will then be read to give you the protein. Okay. Does everyone? Does it make sense? Everyone? Any
Yes,
question?
sir.
Okay. So please remember, the template strand is also called as the antisense and non-coding strand, and the non-template strand is actually called as the sense and coding strand. Okay. So where initiation begins? Where initiation begins? Let us say it began here. This would be called +1. Okay. But when this is +1, actually this strand is being read, and when this is being read, I'll get T, okay, I'll get U, A, and G as part of the RNA. Okay, so U, A, and G. Okay, so remember that even though we say +1, at that point +1, this strand is not going to be read. It's the other strand which is going to be read. Okay. Now, you also have to imagine for the moment that when I have DNA, which is double stranded like this, if I flip the DNA, okay, that means this 5' end now becomes this here, and this 3' end ends up here, then I can actually make another RNA by reading the exact other strand as the template. And when that happens, when the RNA polymerase is going to read this and make the mRNA, then at that time this will end up become the template strand, and this will end up becoming the non-template. So this template and non-template is not absolute definition. The definition of what is template and non-template is always with reference to what the RNA polymerase is doing. So if it is reading this strand, then this becomes template. If it is reading this strand, then that will become the template, and vice versa. Okay, so is that clear? The same sequence of DNA can be read from the other side also. When it is read from the other side, then the nomenclature of the strands will suddenly change. What are the template strand will now become non-template. Okay, does that make sense, everyone?
Yes, sir. Yes, sir.
Okay. So, as I said, initiation involves the interaction of RNA polymerase with DNA at what are called as specific sites or sequences, which are called as Pribnow box or Tata box, and what we call as the -35 TTGA CA sequence. Okay, and that is what we call as a promoter. Okay, now promoter, as they say, are characteristic sequences that direct the polymerase to initiate the transcription, and usually they are located on what is called as a coding strand. Okay, that is the convention. Whenever we talk about where the promoters are, it's always with reference to the coding strand. Okay, and prokaryotes have promoters which are fairly simple. Okay, certain sequences which are always seen near what is called as the start codon, which is the AUG. Okay, so this is actually what happens. So this is what is called the coding strand up. This is the template strand or the non-coding strand. This sequence here is called as upstream sequences. All the sequences down here are called as downstream. All downstream sequences get numbers +1, +2, +3. Okay, depending on where they are from the start site. And all the bases which are present on the upstream part get numbers -1, -2. Okay, so using that convention, you have to realize now that the Tata box or the Pribnow box or the Tata Gaka sequence or -35 region are all upstream. They are all upstream from the initiation start point. Okay, and this one is about 10 upstream and this is about 35 upstream. Okay. Now, when RNA polymerase comes in along with this sigma protein, if you remember that sigma protein ka role is what? To get things initiated. Okay, remember that sigma has only got one function. Okay,
Sigma
to
subunit.
recognize, yeah, initiation sites and recognize them. Okay, so it is needed in the initial phase when the RNA polymerase is searching where to start things. This sigma subunit helps find the -35 promoter and the -10 what is called the Pribna or Tata box. It comes here, it sits like that, and once it sits like that, then see what it does. It starts to separate the DNA, and the way it separates the DNA in such a way that now this portion is separated and my RNA polymerase can come and it can start reading this strand. This is a 3' 5' orientation, and it will start reading this as a template and start making the complementary RNA. Okay, and that is shown in the next slide. Okay, now when the DNA is double stranded like this and nothing is happening, it is called as a closed complex. And when RNA polymerase and the sigma have bound to it, opened it up, and then now the DNA is separated and ready for what is called as transcription, it is called as the open complex. Now the interesting part is once this happens, this sigma just goes away. Okay, it is no longer present. Once it does this function of helping the RNA polymerase find the sequences which are -35 and -10 and open up the DNA, then it disappears. Okay, and now RNA polymerase starts. So see, it starts reading this and it starts to add the RNA. Okay, and the way it adds the RNA is it takes one triphosphate and another triphosphate, and these two are joined with release of pyrophosphate. Okay, and this is where this transcription bubble starts to move, and as it starts to move, it starts to make the RNA. Okay, and as I said, at this point the sigma factor goes away. Now the elongation step is very straightforward. This keeps on continuing. Okay, so the bubble keeps on moving. DNA is unwound to two single strands, and again this is read as a template. RNA is polymerized, and as the RNA is being polymerized, the RNA starts to come out of this RNA polymerase, what is called as transcription bubble. Okay, and at one point you can see actually three things at one time. You can see your double-stranded DNA because see, I separate this, but once I make this RNA and the bubble moves forward, then the DNA again closes up. Okay, so I see actually three strands now. A double-stranded DNA to which seems to be hanging a single-stranded RNA, and very interestingly, some of this RNA is outside of the RNA polymerase, some is inside. Okay, as I say, at any given time, when transcription is occurring, about 10 to 12 to 15 base pairs are always present as part of the RNA complex. And anything more than that, it starts to come out of the RNA polymerase complex. Okay, and this is shown here even more. Here is a double-stranded DNA. It is being unwound. This strand is kept separate so that it doesn't interfere. Which strand is this? This is called the coding strand. This is the template strand. This is being read by the RNA polymerase, and RNA is being made in the 5' to 3' direction. So as this goes forward, this DNA will be separated, the RNA will continue to be made. This is called as the active site of RNA polymerase. This is the DNA which is still to be unwound. It is being unwound in preparation of the RNA polymerase coming in. And this is what we call as the direction of transcription. Okay, as I told you, this is the RNA DNA hybrid that you see inside the complex. And I told you it is anywhere from, as I say here, it shows as 8, but it could be 10 to 12 also. And then as the polymerase moves forward, the RNA is now separated and comes out of the RNA polymerase. And this DNA is again base paired with this DNA, and it goes back to being double stranded. Okay. Now, as I said, this RNA polymerase has got decent accuracy. It's not that it makes a lot of errors, but the error rate is higher than your DNA polymerase. If you remember, your DNA polymerase used to make one error in close to 10 raised to 10 to 10 raised to 12 bases. Okay, so what you call as a trillion bases. Here the error rate is 1 in 10,000. That means relative to DNA polymerase, it appears this is a very bad enzyme. But 1 in 10,000 is actually pretty good, because many times the RNA that you are making are not that long. Okay, so errors will not occur as often as you think. Okay, as I said, even this error rate is okay because we make many transcripts for each gene. So if there is an error, it will be in one of the transcripts. Okay. Now the RNA polymerase works at a rate of about 20 to 30 bases per second, and typically whenever there are GC rich regions, it slows down because it's more difficult to unwind DNA there, and in AT rich regions it always goes faster. Okay. Now see, topoisomerases precede and follow the polymerase to relieve the supercoiling and to induce what is called as recoiling. Okay. Now remember, as the polymerase is moving, the DNA in front has to be unwound. So when the DNA has to be unwound, you have to use topoisomerase in one way, and then after polymerization is done and then the DNA is left, then it has to be recoiled back. So the topoisomerase will work in the exact opposite way, and the DNA will be back to its normal after the polymerase has passed through that region. Okay. So now as this is yeah.
Sir, as the DNA unwind, does the helicase also comes into picture, or it is only the RNA polymerase who does it?
No. That is what I'm saying. This topoisomerase and helicase, okay, they kind of work together in the front and the back. Okay, RNA polymerase doesn't have too much of what is called as helicase activity. Okay, except for that initial part, where that sigma factor comes in. Okay, you recognize the promoter and separate the double-stranded DNA. After that point, RNA polymerase itself doesn't do a lot of helicase activity. It is usually reserved to this topoisomerases. Okay, they will break the DNA, unwind it a little bit, and then allow the polymerase to come through. And when the polymerase passes, they come back again cut the DNA and do the exact opposite, and the DNA is back to its stable structure. Okay, is that clear? So polymerase itself doesn't have too much of helicase activity. It is done by usually topoisomerases, and some cases helicases and topoisomerases. Okay, is that clear?
Yes, sir. Yes.
Okay. So now as this is occurring, and the DNA polymerase, or I should say RNA polymerase, starts to move along the DNA, separating it. At some point, it will reach those sites where the signals are there. KRI, you need to stop the transcription. Okay, and it is these regions of the DNA which are signals which are referred to as stop signals. Okay, Now, in prokaryotes, you have what is called as two types of termination signals. Okay, one type of termination signal relies on a protein factor which is called as a Rho protein, and this is part of what is called as a termination which is Rho dependent. Okay, and there are other types of termination signals which are such that they are self-determined and they cause termination without the involvement of this protein factor called Rho, and that is referred to as Rho independent termination and Rho independent termination signals. Okay, so we have prokaryotic RNA stoppage occurring in two ways. One which relies on this protein called Rho, which is called as Rho dependent termination, and the other which is independent of the requirement for this protein Rho, which is called as Rho independent termination. Okay. Now, what are these two? Okay, now see what happens. Uh, in Rho dependent termination, what is seen is there is a signal which is given by a sequence on the template. Okay, which is the DNA which is being actually read as a template, which is about 40 base pairs in length, and they tend to be what are called as inverted repeats or what are called as hyphenated repeats. Okay, inverted repeats. If you remember, I told you what are inverted repeats. Kind of palindromic sequences, remember, or hyphenated repeats within the same sequence separated by a few bases and again repeated. Such sequences are usually recognized by this Rho factor. Okay, now what is Rho? Rho is actually a ATP dependent RNA stimulated helicase, okay, which binds to these signals. Okay, and when it binds to the signals, RNA polymerase cannot move further. Okay, and therefore when this Rho is present, RNA polymerase simply has to leave the DNA and dissociate from the DNA, and when it does so, it also releases the RNA which is present as a RNA DNA complex inside the polymerase, and therefore now I get what is called my nascent RNA. Okay. Now figuratively, this is actually what happens. As DNA is being separated, okay, as DNA being separated, and one of the DNA strands is being read as a template by the polymerase, and polymerase is making this RNA in the 5' to 3' direction. In those genes where termination is Rho dependent, the Rho protein starts to bind to this newly made mRNA. Okay, and when it's made newly made mRNA, it starts to move. Okay, when it starts to move, it tries to try to catch up to this DNA polymerase. Okay, now DNA polymerase, because it has got a head start, will always be ahead of this what is called as Rho factor. But what happens is, in those genes where you have got this Rho dependent termination, you also have near the what is called as termination sites GC rich regions, and also regions which are what is called as palindromic or have got hyphenated repeats, but these also tend to have a higher GC content. And when there is a higher GC content, RNA polymerase slows down. When RNA polymerase starts to slow down, this Rho catches up. And if Rho catches up with the DNA polymerase, then invariably what it does to the DNA polymerase, I shouldn't say DNA, RNA polymerase, is that it makes the interaction of the RNA polymerase and the DNA, which it is holding as a template, unfavorable. So RNA polymerase simply leaves this complex, and when it leaves the complex, this RNA DNA interaction is broken down and the RNA is released. And when the RNA is released, I get this nascent RNA. At the same time, Rho also leaves. Okay, so I got now my RNA polymerase, my Rho factor, my original DNA which has gone back to double-stranded DNA, and my made a nascent or newly made RNA. Okay, so Rho dependent transcription always depends on the protein Rho and the presence of a DNA sequence which will slow down the RNA polymerase. Okay, so again, when transcription begins and the RNA strands comes out of this RNA polymerase complex, the Rho factor starts to bind to it and in an ATP dependent manner starts to move. It moves along this RNA, and if there is nothing to slow down this RNA polymerase, it will never catch up with the RNA polymerase. But luckily, in those DNAs which use this system for what is called as stoppage of termination, there are always GC-rich regions near the termination site, which slows down the RNA polymerase, allows the Rho factor to catch up with the RNA polymerase, and when it catches up, it dissociates the RNA polymerase from the DNA-RNA strand, again giving you back your double-stranded DNA, polymerase, Rho factor, and the newly made RNA. Okay? Now, the other system, which is called as Rho independent termination, here again you find something very interesting. At the point where termination is going to occur, there is what is called the palindromic GC-rich region, which is followed by a long sequence of AT-rich sequences. Okay. Now, what happens is, because I have got a palindromic GC-rich region, the RNA transcript which is produced is also palindromic, because it is actually complementary to this, right? Because of the self-complementary, and because of this, what you get is formation of what is called as hairpin. Okay, so I'll show that to you. When I have a sequence of what are called inverted repeats, and my RNA polymerase starts to come here and it reads one of the strands and makes a complementary strand. Essentially, the complementary strand will also have what is called as the inverted repeat, and when it has the inverted repeat, see what happens? It forms a hairpin loop. Okay. And this is what we call as a stem loop structure, which is because of the presence of this GC rich region. Okay. Now see what happens because of that. The second feature is also a highly conserved string of A in the template, which are transcribed to U. Okay. So because of this, see what happens when the transcription is occurring. Here I have got an inverted repeat, and then there is a string of what is called as A's in what is called the template strand. So when RNA polymerase comes in and it starts reads this strand, it will first read this, then it will read this, it will make these two sequences which are palindromic. So I get this, then it translates this A and I get a whole bunch of actually U's. Okay, because when RNA is made and A is read, I end up getting a U. So now I got this very unique structure: RNA, then a stem loop structure, and then a string of U's. Okay, and when this happens, typically the RNA polymerase leaves the RNA. It tells it you have reached the stop site. Okay, and you look. Okay. So there are two ways to end what is called as transcription. As I said, one is you take the assistance of what are called as the Rho factor, and the second is you do it without any assistance of this Rho factor by using what are called as specific sequences. Okay. Now, this is what happens in prokaryote. But what happens in eukaryote? Okay. It is in a sense a little more complicated, but then the complexity is more in terms of more exquisite control. Okay. And the way you have got more exquisite control over this process is that you don't use a single RNA polymerase. So when you want to make a specific RNA, you use a specific polymerase. If you remember, RNA polymerase is 1, 2, and 3. The second is as opposed to prokaryotic systems, eukaryotic systems have a requirement of more transcription factors, okay, and more what is called as initiation factors. They also have, in addition to just simple promoters, enhancers, silencers, etc., so that I have got much more control over the rate of transcription than I would see in maybe a bacterial system, where it is mostly, you know, I just get it started or I get it stopped. Here I can have levels where I can have, you know, twofold increase if I want. I have a certain type of enhancer. Then if that enhancer has got also what are called as activator proteins bound, then suddenly the transcription becomes from twofold to tenfold, and so on and so forth. So there is much more control in terms of how transcription occurs, most in both in the positive sense and the negative sense. So I can both enhance transcription as well as silence transcription by making sure I use these sequences and proteins and factors. Okay, much of this is also not understood completely. Okay, so but they know that the controls in eukaryotic systems are more difficult or more complicated than what you see typical in bacterial systems. Okay. No, as I said, RNA polymerase 1, that is the one which makes what is called as typically 28 sRNA, 18 sRNA, 5.8 sRNA. RNA polymerase 2 is the one which actually makes our mRNA, okay, which is later translated into protein. And many times it is made as a heteronuclear RNA, or what is then converted to small nuclear RNA. And then polymerase 3 actually does tRNA synthesis and also synthesis of the small ribosomal RNA, what is called the 5S RNA. Okay. Now there is a toxin called amanitin. Okay. Anybody heard of this amanitin by any chance? Any one of you heard of this? Anyone? Amanitin by any chance? So, before we go further, look at what amanitin does. It very specifically inhibits this RNA polymerase, which means what? If I give you amanitin or introduce amanitin into you, okay, either orally or by injection, what do you expect to happen? Mainly, what do you think is going to happen?
The transcription process would be hindered.
Hindered, but which transcription process? I'm not going to affect too much of ribosomal,
The
and I'm not
mRNA
going to
what?
affect
mRNA.
Which, so if mRNA is affected, then what will be the consequence of that? What will be the result that you will see as far as the
No
cell is
protein.
concerned?
So
I will not be, protein production will be stopped, correct?
translation would be stopped. Yeah.
Yeah.
Yes.
It's not that translation is stopped. There is nothing to do translation with. Okay. So see, the use of terminology is very important. When I say translation is stopped, it might appear that I am affecting translation. Okay. But you have to say correctly that there will be no more mRNA, and if there is no more mRNA, there will be no production of protein. In other words, translation, even if it wanted to, cannot occur. Okay, is that clear? So I'm not giving only mRNA for
Yes, sir.
translation to occur. Okay. Now this amanitin is present in a fungus which is called as Amanita phalloides. Amanita phalloides. Okay, Amanita phalloides is actually a mushroom. Okay, so it's very interesting, and I don't know if you know of this. Okay,
So it is
uh,
cholinergic blocker, I suppose.
it is actually not a cholinergic blocker because I already told you what it does. What does it do? It inhibits RNA polymerase 2 and it prevents protein synthesis. Okay,
No, it is
so
related to mushroom poisoning, something.
correct. Mushroom poisoning mainly occurs because of effects on protein synthesis, very little because of cholinergic. Okay. Okay,
Okay,
cholinergic
sir.
poisoning occurs because of consumption of other hallucinogens. Okay, for example, things like consumption of, let us say, atropine, okay, or strychnine or brucine, something like that. Okay, or too much nicotine. Okay, if you overdose on nicotine or something like that, then that affects the cholinergic system. Amanitin works basically by inhibiting protein synthesis. Okay. Now, lot of, I don't know whether you know about this, but there's a terminology which teenagers use, which is called a shrooming. Okay, have you heard of this?
Yes,
It's
sir.
called shrooming. So, what is shrooming? Anyone know what is shrooming? By any chance? Okay. So, this is where you get together and just like we go trekking, no? कि हम उन waterfall देखने जाते हैं या अपन fort को देखने जाते हैं या let us go to
So it's like
a
mushroom,
resort. हाँ,
mushroom collection, something like that.
correct. It is collecting mushrooms.
Hunting
हाँ,
for mushroom.
correct, correct. But the idea of collecting mushrooms when you do shrooming is not to consume it as a diet. Okay, because we also eat mushrooms, right? Mushroom soup, वगैरह. When we say I'm doing shrooming, the idea is to go collect mushrooms with the intention of collecting those mushrooms which when you consume will give you what are called as hallucinogenic effects or what you call as psychedelic effects or what you call as euphoric effects. Okay, and there are certain mushrooms which do that. Okay, where if you eat mushrooms, no, rather than it giving you, food and protein, it actually has toxicants which will cause what is called a hallucination. Okay, and certain mushrooms do that to you. But you have to always be careful that certain mushrooms are also what are called as killers. If you eat them, you will simply die because of them doing something like this. For example, if I consume Amanita phalloides, then if I am not careful, I'll basically stop protein synthesis. And when I stop protein synthesis, my cells will start dying. Okay, and basically it will start doing what is called as tissue damage, and my organ failures will start to occur. Okay, so I have to be very, very careful. And the worst part is, the more toxic the mushroom, the more attractive it tends to be. Okay, so if you are going shrooming, no, the first mushroom you will spot will be the ones which are nicely colored or which have got designs on them, and you'll get attracted to this. And animals also tend to get attracted to these mushrooms. And these mushrooms are very dangerous because the more colorful they are, the more toxic they tend to be. Okay, and the ones which you consume as food are the blandest ones. Actually, घर में जो मशरूम लेके आते हैं ना, उसको देखोगे तो उसमें कुछ भी ऐसा कलरफुल नहीं लग रहा है. It looks very, very bland. And invariably, that is how the correlation appears to be. The blander the mushroom, the more safe it is. And the more colorful the mushroom, the more dangerous it is. Okay. So this Amanita phalloides.
Appearances are deceptive.
Very, very deceptive. Okay. So if you are, down the road, if you are going to go look for mushrooms and all, be very, very careful. Okay. Just a second. Okay, so be very careful. If you consume the wrong mushroom, you will be done. Okay, it is very, very toxic. Okay, now, so that is one aspect of eukaryotic transcription, is the fact that the promoters which are present are located slightly differently than what you see for eukaryotes. Okay. So if you remember eukaryotes, we talked about the Tata box being also called as the Fribnov box. And where was that present? Which region?
Which
region? -10. Then we also talked about, Tata Gaka. Okay, TGCA.
-35.
It was
-35.
-35. So here in eukaryotes, what you find is there are two sequences which are important for transcription. One which is very similar to the Tata box, which is actually called as a Hogness box, okay, which is actually located -20 to -30. And the other, which is equivalent to what is called as a TT Gaka box, or the TT Gaka region, that is here called as a CAT box, CAAT box, and that is located about -70 to -80. Okay. Now look at that also. In addition to that, see this, there can be regulatory elements, there could be enhancers, silencers, which are further upstream, and these also play a role as to how transcription will occur. Okay. Now, further, see what happens in eukaryotes. Sometimes these regulatory elements are also present after the transcription unit. Okay. When we say the transcription unit, this is where the gene is. Okay. So this will be the +1 and this will be the end of the transcription. So sometimes regulatory elements are also present downstream. This is very unusual. Okay, in prokaryotic systems, you don't have this complex regulation of a gene, where a gene is regulated by sequences which are present very upstream on the 5' end, and they are also present downstream, which is on the 3' part after the gene is there. But they all play a role in how a eukaryotic promoter will be controlled. Okay, so there are two differences which you have to remember. One, the fact that different polymerases are involved in making different types of RNA, and the second is that the promoter and the promoter architecture is slightly different, where the Tata box or the Fribnov box is called as the Hogness box or Hogness box. Is located -25 to -30, and rather than having a TT-GACA sequence at the -35 region, we have a CAT box, what is called a CAAT box, where this sequence is seen, okay, which is at -70 to -80. And not only that, in addition to that, we have enhancers, silencers and regulatory elements which could be present upstream or which could be present downstream of the region which is actually going to be transcribed. Okay. Now, in addition to these two differences, in case of eukaryotes, after transcription is over also, the RNA that you get is not ready for making the protein or not ready to do its function. Okay. So many times in eukaryotes, you get an RNA which is referred to as a primary transcript, which actually needs to be modified to become what is called functional. And this functional part could be the tRNAs which are being produced, the rRNAs which are being produced, or the mRNAs which are being produced. Okay. Now, sometimes these primary transcripts of mRNA are also referred to as heterogeneous nuclear RNA or hnRNA, because they are actually present in the nucleus. That is where the modification is going to be occurred, and then once they are converted to what is called as mature RNA, which is now called mRNA, they are released into the cytosol. Okay. Now, please remember that in prokaryotes also you have post-transcriptional modification. But in prokaryotes, the post-transcriptional modifications are reserved to generation of only tRNAs and rRNAs and not for generation of mRNA. So that is a big difference. So in both prokaryotes and eukaryotes, when you get a primary transcript related to what is called as tRNA, it is going to be subject to post-transcriptional modification. Likewise, both in prokaryotes and in eukaryotes, when you get a primary transcript related to what is called as ribosomal RNA, there also it is going to be subject to post-transcriptional modification. But when you talk about a primary transcript related to what is called as a messenger RNA, then typically in prokaryotes, there is nothing called as a post-transcriptional modification, but in eukaryotes, that mRNA or what is called as heterogeneous nuclear RNA is subject to a lot of changes before it becomes ready for translation, and that change results in what is now called as a mature mRNA or matured messenger RNA, and that is a feature of only eukaryotes. Okay, so tRNA production by PTM and rRNA production by PTM. PTM stands for post-transcriptional modification, occurs both in prokaryotes as well as in eukaryotes. But PTM of mRNA, which is involved in giving you protein, occurs only in eukaryotes. And what are these changes that occur after transcription to this mRNA, which is very unique to eukaryotes? So these are the changes. Okay, one which is called as RNA splicing, and many of you have heard of this. Okay, because you already have told me when I asked questions earlier in rDNA technology, the concept of what is called as introns and exons. Okay, so most of you are familiar with that. We also talked about the poly-A tail. Remember when we talked about poly-A tail? Anyone recall when we discussed poly-A tail?
Yes, sir.
When we did cDNA technology, remember copy DNA technology or
Yes, sir.
copy DNA library or complementary DNA library. And there are other two things which I have not mentioned earlier, which are also here, which is addition of what is called as a 5' cap and what is referred to as RNA editing. So we're going to look at all four of these one by one. Okay. So the first part I want to look at is splicing. Okay. Now in what is called as eukaryotic systems, it is very common that when DNA is read to give you what is called RNA transcript, which later on is going to be coding for a protein, then the primary transcript that you get is relatively large with respect to the protein it is supposed to code for. Okay, for example, the other day, if you remember when we talked about insulin, we said that the final functional insulin has got only 51 amino acids. So if that is the case, the DNA should have been at the most 153 bases. But if you recall how big the gene for insulin was, it was about 1400 bases. Okay, which means there's a lot of RNA which is being produced by reading the DNA, which later has to be removed because it actually doesn't code for any protein. Okay. So what this means is that the genes which are actually there, which gives you finally a protein, has got parts which are actually what referred to as coding parts and non-coding parts. Coding parts are those which finally end up as being read and actually giving you the protein, and the non-coding parts are the parts which apparently are part of the gene, but later on you never see the effect of these non-coding parts in the actual protein. Okay. So for example, let us say I have a gene like this, which is 7700 base pairs, which starts from here and it ends here. I might find that these regions which are there, A, B, C, D, E, F, G, they are there as part of the gene, but they do not code for anything. The actual code here is the sequence which are present as part of 1, 2, 3, 4, 5, 6 and 7. So what this means is, when I make the RNA by reading this DNA, I have to then remove these sequences which are not needed. Okay, and these sequences are referred to as intervening sequences, or what are called as introns, and they are removed when the RNA is being what is called as modified to give you what is called as mature RNA. Okay, okay, that is also shown here more correctly. So I have a DNA as a long sequence. This is coding for, this is called as a gene for a given protein, and in that gene there are these regions which are actually important, which are shown in colored. They are called as exons. Then these sequences here, okay, in the middle, which are called as introns, are the ones which are actually non-coding. They are not going to be part of the final protein. So when this DNA is read and I get an RNA, my RNA is going to be very long, and during the process of what is called as PTM, or post transcriptional modification, and what is called as splicing, I splice out this intron, I splice out this intron, I splice out this one. That means I simply cut it out, and after cutting it out, I join the remaining one. So I join the blue to the green, to the red, to the light blue, to the yellow, and I finally get what is called as a mature RNA. Okay, so this splicing is one of the modifications that occurs, and is very unique to what is called as eukaryotic maturation of messenger RNA. Okay, now this process of what is called as splicing is very unique because it doesn't actually use many times any proteins. The function of splicing is done by the RNA itself. Okay, and this was the first time it was shown that RNA itself has got catalytic activity. Okay, and this is where if you were to look at how biochemistry was taught maybe about 40-50 years ago, at that time people used to make a statement that all enzymes are proteins. Okay, and then as soon as this was discovered, then we started making a statement, enzymes are mostly proteins, but sometimes it can be also RNA. So the first example of a catalytic RNA was actually RNA which does what is called as self-splicing. That means it removes itself. Okay, so it is part of an exon, I should say it is part of an intron which self-removes itself. Okay, and it does so by forming, what is this typical lariat, or what is it called. Okay, now these are all mechanisms of splicing which probably are not important for you, but I just put it here so that if in case you are interested, you can look at it, how this occurs. Okay, now the second part is what is called as 5' capping. Okay, now as part of what is called as modifying the RNA after transcription, or as part of what is called as PTM, post-transcriptional modification, in eukaryotes, a very unique cap is also put at the 5' end of the RNA that is made. Okay, and for this, what is used is a very interesting nucleotide, which is 7-methylguanosine. Okay, 7-methyl means methyl is present on the nitrogen, which is numbered 7. Okay. Now, the way this is done is that it's better to just look at it and see what is done. So, if you see, this is the RNA. This is the RNA, and this is the 5' end. The 5' end is CH2OH. So, this 5' end is added this methyl guanosine nucleotide. So, see this methyl on the 7th position. This is a base which is guanosine. This is the sugar, okay, and this is the 2 phosphates. Okay, actually, originally it was a triphosphate, but out of the triphosphate, 2 phosphates are retained, and this is a phosphate at the 5' end of this RNA, and they are joined together to give you a very interesting 5' triphosphate bridge. Okay. See, normally we have always seen as 3' being attached to 5', 3' being attached to 5' as part of the links which you always look at. This is the first time you are actually seeing 2 5' ends are attached to each other through a phosphate. Okay, so that's why it is called as a 5' triphosphate bridge. Okay, the other thing I also wanted to notice is not only this methyl group is used to methylate the nitrogen of this cap, which is a guanine, the first 1 or 2 bases of the RNA are also sometimes methylated. Okay, so this is part of what is called the 5' end modification, which some people just call as 5' end capping. Okay, the same is shown here. So again, this is that RNA, this is the 5' end. Again, you see this 5' end ka and this 5' end ka connection through 3 phosphates, and this is that guanine which has got a methyl group. Okay. Now, what is not shown here? What was not shown here is that this also can be methylated, and this is more shown here. See this methyl group and this methyl group. So the first 2 bases ka O, which is of the RNA, are also sometimes methylated as part of this PTM, or what is called as post-transcriptional modification. Okay. Now the third modification is what is called as addition of a poly A tail. Okay. So as I remove the introns, as I put this 5' methyl guanosine cap, I do another modification where at the 3' end of the RNA, a string of anywhere from 40 to 200 adenines are added in a template independent fashion. That means what? There is no template here. Remember, RNA has already been made, it has separated from the DNA, and the RNA polymerase has left. So now, when I do this poly A tailing, I do not have anything to, as a template, there's a enzyme which specifically comes in, which is called as a polynucleotide kinase, which will add bases, adenine, to the 3' end, and it does so without a template. And because it does so without a template, there's always a variable number of A's which are added, and this variable numbers can go from anywhere from 40 to 200. Okay. If this was added in a template dependent fashion, then this number would have been very exact and would have matched how much ever, let us say, T's are there on the template, because every time a T is there, my polymerase is going to add or kinase will add a or okay. So here, because there is no template, what happens is I have this cleavage. Okay, remember I have got this polymerase adds to the 3' end. Okay, so this has come out from the RNA polymerase. Okay, and then I am sitting there, I put a cap, I have removed the exons, then I just simply add a tail. Okay, everyone clear? Now the last part, which is what is called as RNA editing. Okay, this is something which is a little more tricky. Okay, and it says RNA editing is a molecular process through which some cells can make very discrete changes to specific nucleotide sequences within an RNA after it has been generated by RNA polymerase. In fact, more correctly, I should say after it had already been subject to 5' capping, what is called as intron removal and poly A tailing. So this is over and above that. Okay, and it seems to occur in most living organisms, and is, as I say, one of the conserved properties of RNA. Okay, and this is a very important statement: evolutionarily conserved properties of RNA. Okay, so this ability to do RNA editing or RNA modification is something which appears to be a property of some of the specific RNAs which are present in living organisms. Okay. Now, what this is and how this occurs, maybe we will reserve that for next time. Okay. So, I am going to stop here for now, and I'll stop my sharing, and let us see if there are any questions at this point. Okay. Let us see. Let me go to my chat box here. Okay. One of the questions. Let me. Can it be the initiator site is a -10 block itself? No. Shivam is asking, does initiation occur at the -10 itself? No. Relative to -10, initiation occurs around 10 bases away, which is called as a +1 site. Rutuja, can this process be stopped purposely by poison or something? Yes, it can actually. You can have compounds, if you design correctly, can work at the level of the promoter, and when they bind to the promoter, they actually stop the process of transcription. Okay, yeah, definitely you can. Okay. Third, again Rutuja, can instead of Tata we have AAAA or TTTT, as the number of bonds will be the same if you have ATTA. Yeah, technically yes. Okay, but what has been observed in nature is that we rarely have sequences where one strand is completely A and the complementary strand is completely T. All the examples that we find now in terms of looking at DNA based on where transcription appears to begin from, we find the sequences tend to be a mix, okay, or Tata. Okay, so no a specific reason for not having A or T. That would have worked equally well, but nature as of now appears to follow this TATA. Okay. Why the hairpin termination is found in eukaryotes? Okay, see when you form an hairpin, what happens is suddenly the structural changes that occur because of hairpin and what is later on a poly U. Okay, they change the dynamics of how the RNA interacts with RNA polymerase and how the RNA interacts with the DNA also. Okay, and that promotes the dissociation of the RNA polymerase from the DNA. Okay, in RNA polymerase, how does the alpha subunit identify which DNA sequence is to be transcribed? Okay, now I've told you in a very simple fashion the way things work, but remember there are also what are called as transcription factors which play a role. There are also proteins which sit on what is called as the promoter sequences or repressor sequences, and they all have some affinity for RNA polymerase. Okay, and they allow the RNA polymerase to bind not only to them, but after binding to them, modulate the activity of polymerase in terms of enhancing its affinity to start transcription or slowing the RNA polymerase down. Okay, and many of these interactions are based on what is called as protein-protein interactions. Okay, so the repressors and promoters proteins which sit on the sequences are able to recognize certain regions of the polymerase, because polymerase is a globular entity, right? You have alpha, alpha, beta, and your holoenzyme and sigma factor. All of these have a three-dimensional shape, which is actually part of the interaction with proteins which bind to the promoters and also the promoter sequences themselves. Okay, and that is how the polymerase is able to identify the sequence which is to be transcribed. Okay, you will find that in lot of places in the DNA where there is nothing to do with a gene, these sequences will not occur at random, and there will be a lot of these repeats and short sequences which are not making sense, or a sequence which has got lot of stop codons. Okay, so okay, you cannot make a protein if there are a stop codon every 20 to 30 bases along the DNA. Okay, so generally regions where DNA codes for something, you'll find a long sequence many times without any stop codons. Okay, that tells in a way your machinery. Okay, yeah, here is where there is a sequence which probably could code for a protein because I have a whole length of bases uninterrupted by what is called as a stop codon. Okay, and that sometimes is part of the identity. Okay, now this what I am telling you at the third year B Pharm level is a very simplistic view. In fact, if you go to any little advanced text on biochemistry, new chapters are written on how the interaction of polymerase occurs with DNA, how the interaction of polymerase occurs with the factors, protein factors, and how this interaction seem to affect how the polymerase recognizes where the transcription should begin. Okay, and again this second issue: why is there a gap between the initiation site and the Tata box? Why can't it be placed together? Again, this is probably because the architecture of the protein is such that I recognize one site, and that recognition site and the initiation site, which has got catalytic activity, are not right next to each other in the protein, and they're separated by a little distance, and that results finally it's like me asking you that when you're cutting tomatoes, no, for making a sandwich, okay, why do you make a cut which is a millimeter or two apart? Why don't you cut the tomato so thin that the slices are fully transparent? I so transparent I do not see even the red color in it. Okay, and then you will tell me, okay, that is because the way I hold the tomato and the way I put the knife, the sharpness of the knife, the fact that I'm afraid that I'm going to cut my finger results in a little gap between where I hold the tomato and the knife so that I don't cut myself. Okay, so something very similar probably happens even in these systems where those sequences which are used to recognize where initiation is supposed to start and the actual catalytic site where bases can come in and they can be polymerized are separated in space, and because of that there is a gap between what is called as the initiation site and the Tata box. Okay, but that is probably why. Okay, does the number of poly A tail nucleotides play a role like stability? I really don't know, but it appears that the tail is variable, but it cannot be too extensive or too short also. Okay, so there is something to this poly A tail that it needs to be a certain length. It can't be too short, it can't be too long. Okay, but what exactly, maybe I have to look up and I'll try and see if I can find anything out on that. Why do we have to add a 5' cap and a poly A tail? Okay, I still can't get this use. Okay, so the reason is when RNA is being made, no, the lifetime of the RNA is very short. Okay, because it is inherently unstable. Okay, and there are in our body also what are called as nucleases, which will start degrading the RNA. Okay, so one of the reasons for 5' capping and poly A tailing is especially in eukaryotes because the RNA now is going to be transported from the nucleus to the cytoplasm, and in the cytoplasm it is going to be translated. So this it is supposed to have a certain lifetime, okay, a certain level of stability because it is going to be subject to transport process, right? So I make the RNA in the nucleus, but translation is not occurring in the nucleus. It is actually occurring in the cytoplasm. So for that time, I have to protect my RNA. Okay. And again, how are introns and exons identified? There are specific sequences in this regions which are called as introns, which tell you where the boundary between the intron and exon is. Okay, so this cutting of introns and exon actually doesn't occur at random. There are certain sequences which are conserved, which tell you that these are the borders between introns and exons, and it is there where the cutting occurs. Okay. So,
Sir,
yeah, go ahead. Hello.
sir, so why couldn't all the exons be together? Why are there introns, inserted in between the exons?
Oh, I am
Why
right.
couldn't the whole?
Okay, okay. Okay, so there are several ways to answer it, and maybe I'll give you one viewpoint. Okay, uh, see, if I have my entire protein ka sequence in one shot, okay, uh, it will help me in one way. It will help me in the sense that my gene will be shorter, correct? So my insulin gene now will be only 151 bases. I am done. Okay. Uh, so in that sense, you might ask, okay, why the question that you're asking is why are 1,400 bases coding for insulin? Because much of the part is not coding for anything. Okay. Now I look at it from another way. If I have a contiguous sequence without any introns and exons, which code for my final protein, then if any damage occurs there, basically I am done. Okay, because the chances are that the damage will result in a protein which is non-functional, and because of that I might have other consequences where it might result in my death, or it may make my life miserable, or make things very unfavorable for me. Okay, so by taking my gene and splitting it and putting lot of sequences in between which are actually not going to be part of the final this thing. Now when random damage occurs, okay, to DNA, then my actual gene is split. Okay, so then the chances of me damaging my functional sequence is a little less, because I've just separated. It's like I'll give you a simple example. Okay, if you have 10 chocolates, okay, and you keep it all in one pocket, okay, and your top pocket, and you come to the canteen, okay, and let us say you're a guy and you keep it in your top pocket, and you're sitting in the canteen, and immediately that chocolate is going to be visible to everyone. Okay, what do you think will happen in about 2 minutes?
All the chocolates are gone.
All the chocolates are gone. And why are they gone? They are visible.
Everyone will, yes.
Everyone will take it and they'll just grab it. Now see what I do. I keep chocolates, 2 on the top pocket, one of my left pant pocket, one of my right pant pocket, one of my back pocket, one of my other back pocket, one actually in my bag, one in my socks. Okay. Now I sit in the canteen. Again somebody spots a chocolate. But now see what happens. At the most 2 will disappear. Okay. And somebody might think I had only 2. Only the intelligent one will ask, और किधर छुपा के रखा है क्या? Okay. So what I have done now is kind of protected myself in a sense. जैसे अपन बस में ट्रैवल करते तभी घर में से एक इंस्ट्रक्शन आता है मालूम मम्मी डैडी से. When you travel by bus or train, your parents instruct you something very important when you leave the house.
Phone bag में.
फोन बैग में रखो. सब पैसे
अलग-अलग
एक
जगह पे रखना.
पे रखो. क्यों अलग अलग जगह पे रखो? क्योंकि किधर से भी उठाया किसी ने पैसा तो तुम कंगाल नहीं हो जाओगे. समझा ना? So maybe during evolution also, because evolution realized that there is a lot of insults occurring to my DNA and my DNA can potentially be damaged. Then what is the best way to design my DNA so that when these damages occur, they do not actually damage what is most needed by me. Okay, and maybe that is also the reason why only 1% of our DNA is coding. Okay, almost 95 to 99% of our DNA is just junk. Okay, so look at the junk. एक तो सब जगह junk है, उसमें gene है, उसके अंदर भी junk है. Okay, so if some damage were to occur by a chemical, chances of the damage occurring on a place where there is no gene is always more than a place where a gene is. Okay, where I shouldn't even say gene, the business part of the gene is less. So it's maybe a way to protect. That is one way. The other answer is this. Okay, now during evolution, there is also evolution where I take things which work very well and I use it again and again. Okay, Abhi, for example, and I put this in question to the ladies in the group. Okay, without being gender biased. Okay, How many of you ladies would like to have your phone ka outer cover? Okay, we put a little cover on the phone, no? उसको क्या बोलते हैं?
Yes.
What is the terminology for that? ऐसे एक प्लास्टिक ढक्कन लेते हैं ना अपन, so that that phone doesn't get damaged by by things falling on it. Except for the screen, everything else is protected. That is what is called phone cover. What do you call it?
Back cover, phone cover.
Yes,
Back
sir.
cover. Okay. How many of you ladies would like the back cover of the phone to match your dress they are wearing on that day? I'm not asking this question to guys because guys generally don't worry about this. But many times a girl, imagine you're wearing a nice pink top, and the phone which you're carrying has got a green back cover. You think, this is not quite right. It would have been so nice if my phone had a back cover which was also kind of a shade of pink. Anyone? Any ladies agree to me?
Yes.
With this all? No. So what do I do? Do I buy like 10 different phones with 10 different back covers? I don't do that. No, I keep the phone the same. All I do is change the back cover. So I buy this set of back covers of different colors. Okay, kind of matching the dress profile that I have, so that on different days all I have to do is simply remove the cover and put another one. This is also like the bindis that you put, okay, or the earrings. Okay, you keep a little set so that it goes with your dress. Okay. Now in evolution also that happens. Okay, if I have got 3 different proteins which are going to use ATP to drive the reaction, okay, then these 3 proteins can take that part of the protein which binds ATP and hydrolyzes it and use it in common with another site where different substrates are able to bind. So you see what I do? I take this ATP binding site which is common to another protein and use it as part of my gene. The only thing I change is the part where my substrate binding site is different. So when in evolution such mixing and matching occurs, then what you find is lot of these exons are common to many proteins, but over time they've evolved and become part of different genes, and that's why they're not right next to an intron. Okay, so I gave you 2 viewpoints of why maybe introns or exons are separated, okay, and not together. Does that make sense to you, Aditi?
Yes, sir.
And there could be many other reasons, okay, but these are 2 nice reasons as I gave you, so that you realize, okay, they are separated, and then second, there are certain sequences which allow you to identify where introns and exons are. Okay, yeah, go ahead, Aditi.
Sir, and in prokaryotes, we have only a single polymerase
Yeah.
which binds for all the 3,
Correct,
mRNA,
correct.
RNA, and T.
Correct.
So how does it like identify when to make mRNA, when to make rRNA?
It doesn't. It doesn't. It actually doesn't identify. It just reads, because I told you the promoter sequences are much simpler in prokaryotes. So whether it's a tRNA or rRNA or mRNA, the way the the Primno box and the TTGC GACA box are set up are very similar to all the different types of RNAs. Okay, while the absolute sequence of what is the Hodgeness box and the -70 and -80 region in a eukaryotic is subtly different, which allows our polymerase to recognize the gene which is going to be downstream. Is it a gene for mRNA? Is it a gene for tRNA? Or is it a gene for rRNA? And then because of that, different polymerases can potentially come and then start the production of mRNA. Okay, we are just a little more complex, both in terms of the sequences which are present upstream and downstream, relative to prokaryotic systems, where there is much less differentiation, so there is need only for one polymerase. Okay, but it doesn't distinguish कि rRNA है कि tRNA. It just goes there, sits whatever उधर से -10 से +10 side से चालू कर देगा, and it will start. It doesn't worry about the product. Okay, is that clear?
Yes, sir. Thank you, sir.
Okay. Any more questions? Did I answer everything which is there on the
Yes, sir.
Anyone whose
Yes,
questions are unanswered? We need more clarification. Anyone? Okay. So I think I answered everything. So can I stop my recording? Stop my recording and